All right. So hello, I'm Jess Mullins. I'm a master's student in Museum and Field Studies at the University of Colorado Boulder. This photo of a bumblebee queen fleeing a thunderstorm nicely sets the stage for my talk today, as I'll be talking about how a flood impacts wild bee communities. This work is in collaboration with Virginia Scott, the CU Museum of Natural History Entomology Collection Manager, Adrian Carper, who you know already, and Dean Bowers, my advisor, a professor at CU Boulder, and the curator of the CU Museum of Natural History's Entomology Collection. Before I get started, I'd like to thank the land managers, the City of Longmont, and Boulder County Parks and Open Space who enthusiastically permitted this research on the land that they manage. Also, this work would not have been possible without immense help in both the field and the lab from collectors, data processors, and bee identification experts. We would particularly like to thank Mary Jane Howell, who has volunteered to work on this project from the very start, through a 500-year flood and now a pandemic. We are extremely grateful for funding from the City of Longmont, Denver Audubon Society's Lois Webster Fund, and the CU Museum of Natural History. According to the 2018 IPCC Special Report, the average global temperature has risen from the pre-industrial era by 1.5 degrees Celsius. This temperature increase has led to changes in weather patterns, bringing more frequent and more severe weather events, specifically to arid regions like Colorado. If the planet continues to warm at its current rate, this will worsen, and it is extremely difficult to predict exactly when these storms will hit. And in turn, little research has been done looking at the impact on biological communities before and after extreme events. In September 2013, an unusual weather pattern brought what many have classified as a 500-year flood to Colorado's Front Range. In some areas, over 16 inches of rain fell in just a few days, which is more precipitation than most areas of the state in a single year. This flood resulted in major rivers and their tributaries breaching, such as two tributaries of the South Platte River, the St. Vrain Creek and the Left-Hand Creek shown on the map. The map shows the city of Longmont, and the normal extent of the creek is shown in dark blue, and the light blue highlights the total area flooded following the storm. The dotted green line illustrates an urban trail, the St. Vrain Greenway. These photos were taken on September 13th, 2013, after most of the rain had fallen, but much of the city of Longmont was still underwater. The markers on the map denote where the photo was taken. Note the brown coloration as water breached from the creeks. The habitat along the water is a riparian corridor, which is rich in floral resources and diverse nesting sites for wild bees. As you can see from the map and the photos, this habitat was heavily impacted by the flood. Most flowering plants, such as those found along the trail, require animal pollination to set new seeds, and most often, this pollinator is a bee. Pollination is crucial in helping recovery following large disturbance events, such as this flood, so that the plant community can reestablish. Most wild bees are ground nesting, and the inundation of this soil could introduce detrimental impacts to the bee nests found along the St. Vrain Creek. In some regions, ground nesting bees are specially adapted to living in floodplains. However, little is known about how non-flood adapted species, like those from Colorado, fare storms. As Dr. Carper mentioned at the beginning of this meeting, pollinators, in particular wild bees, are incredibly diverse. There are over 900 species found in Colorado and about 660 occurring in the Front Range, where our research took place. The bee life history traits vary by family, more finely down to species level, and each of these traits could impact a bee's ability to respond to floods. While there are plenty of life history traits not listed here, we chose 3 main categories to assess how Colorado bees could respond to a flood. Those categories include nest location, whether it is underground or in an above-ground cavity, nest architecture, whether the walls of the nest are lined with a water-resistant substance or not, and sociality, whether a bee is social, solitary, or parasitic. Due to difficulty in classifying whether bees have specialist or generalist diets, that is, feeding on one type versus many types of flowers, we have yet to analyze diet. Now that we know more about some life history traits that could be associated with flood response, let's look at how these traits are dispersed between 5 of the 6 different bee families found in Colorado. First, let's talk about Coletidae. This family contains cellophane and masked bees, all of which are solitary. Masked bees are found nesting in pithy twigs of shrubs, such as sumac or rose, and cellophane bees nest in the ground. They build watertight cellophane linings in their nests from secretions. Sweat bees are the most abundant bee family found in Colorado. If you've ever been out on a hike or gardening and a small bee landed on your arm and didn't sting you, it's probably a sweat bee taking a drink. They nest exclusively in the ground. All of them line their nests, and they can be either social, solitary, or parasitic. The mining bees, as their name suggests, build their nests in the ground. All of them are solitary, and some line nests while others do not. You can catch a sight of the mining bee Andrina in the spring if you stop and take a look at the flowers on willow trees. You'll find their nests at the base of the tree. Bees in the Apidae family are maybe what we are most familiar with. Though not pictured, the introduced European honeybee belongs to this family. Some other familiar members include bumblebees and digger bees. This family covers all life history traits we've assessed. Now for my favorite family, Megachilidae. These bees can live in the ground or in cavities, and some even construct their own nests with pebbles and mud on top of rocks or twigs. All of these bees are solitary, and some species even parasitize others within the family. Now that we understand the vast diversity of wild bees in Colorado, how did we test their response to the flood? Some of this was luck. As part of a pilot bee biodiversity monitoring project, Virginia Scott fortuitously sampled the St. Vrain Greenway 1 year before the flood. This is an area of prime bee habitat that has not been sampled in many decades. The flood introduced a natural experiment, and we resampled in 2014 and 2020 to assess different stages of bee recovery. In each of the sampling years, we visited each sampling site every 2 weeks, beginning in March and ending in October. We hand netted all of the bees we encountered, recorded the plant or substrate upon which it was found, and brought all samples back to the lab. Each bee was pinned and labeled, and then identified to genus using the bee genera of North and Central America key by Michener and colleagues. These bees are vouchered at the CU Museum of Natural History. While time spent in the field is, in my opinion, the most fun and provides an opportunity to learn about bee behavior, the most intensive portion of time spent doing this research is processing. Over 3 years, we spent 300 hours in the field collecting bees. And while it is difficult to quantify the exact numbers, we estimate 10 times this amount of time was spent processing, with identifications being the most arduous. Some bee groups require consultation with very few specialists found worldwide. With a rare opportunity to look at changes in a bee community pre and post-flood, our objectives were to assess the changes in diversity and abundance, both short and long term. We predicted to see a decline in both abundance and diversity from pre-flood to post-flood, and a rebound 7 years post-flood. Next, we wanted to identify life history traits that could provide resilience to a flood. These traits include nest location, nest architecture, and sociality. We predicted bees who nest above ground and those who line their nests would have greater relative abundances than the years following the flood. Additionally, we expected to find lower relative abundances of solitary bees and thus their parasites from pre-flood levels. As we continue to work on species-level identifications for the bees we caught this season, we've begun our analyses looking at the genus level. Research by Van Rijn and colleagues found in 2015 that looking at a broader taxonomic classification, such as genus, provides similar findings to an analysis at higher resolution. We applied this approach to our analysis and found genus richness follows our predictions with near statistical significance. There was a decline in genus richness following the flood, shown in blue, and a recovery in 2020, 7 years following the flood, shown in yellow. We expect as we continue to refine our data set, the significance will greaten. Next, we assessed changes in relative bee abundance by family for each of the sampling years, as this can provide clues into which life history traits could lead to resilience. 1 year following the flood, The community was made up of 60% sweat bees. In 1986, bee taxonomist George Eichort noted that bees from the largest group within the sweat bee family, Lasioglossum, they live in disturbed habitats and visit a wide variety of plants for pollen and nectar. It is possible the disturbance from the flood provided new nesting habitat for these bees. The only family's relative abundance to remain below pre-flood levels is the cellophane bee family. One group within this family, the masked bee, builds their nests in pithy twigs. Perhaps these nests were broken and washed away, and similar habitat has yet to regrow. All other groups' relative abundance has returned to similar levels of pre-flood surveys. In analyzing bees based on sociality, we did not classify members of the sweat bee genus Lasioglossum, as species in this group can be assigned to all three categories. Sheffield and colleagues found in 2013 that parasitic bees can be an indicator of an intact bee community. Our recovery year surveys, shown in yellow, resulted in the lowest relative abundance of parasitic bees. These bees parasitize mostly solitary bees. If solitary bees are in decline in a community, this will be shown in the relative abundance of parasites. In future analyses, we will assess changes in specific parasitic bees and their hosts, for example, the cuckoo bee nomada and the mining bee andrena over the 3 study years. We did find more social bees this year as expected, likely due to established reproductives such as bumblebee queens. Contrary to our expectations, we did not find significant changes to the relative abundance of bees based on nesting location. The bees who nested in the ground either fared the flood well or reestablished on the greenway from nearby unimpacted areas. And very few bees do not line their nest at all, and we cannot make any definitive conclusions based on the presence of nest lining as a resilience predictor. Here it is also important to note that experts think bees line their nests to maintain humidity within the nest cell. This is important in deserts where the bees are most diverse. The decline in relative abundance of bees who line their nests with cellophane is likely due to nest location and not lining. In the figure showing bee relative abundance response by family, we saw a similar pattern. As mentioned, the twigs that the masked bees nest in have not reestablished following the flood. Preliminary analyses show that perhaps floods do not impact bees as severely as it may intuitively seem, and we've measured a return of bee richness to the same levels found before the flood. Similar to many other biological systems, moderately disturbed systems are the most diverse. It is the frequency and severity of future disturbances that may introduce problems. For wild bees and other plants and wildlife to recover from disturbances, it is important for them to have a greater area of habitat available. This way, there is more area that could remain unimpacted by a disturbance, and then the plants and animals can recolonize from there. In future months, we will incorporate floral data to assess changes in the floral network, look at resilience in bees based on their diet, and continue obtaining species-level identifications to understand the flood impacts on a species level. Thanks for tuning in today.
Awesome. Thanks so much for that, Jess. I mean, again, more tremendous natural history, right? I mean, how long did it take to come up with some of the classifications for whether or not a bee lines itself with a Dufour gland secretion, right? I'm assuming those papers are kind of hard to come by, right?
You know, I got really lucky because the year that my master's started, the Solitary Bees book by Danforth et al. Came out, and there's a table that has done this extensive lit review. And it made it a lot easier to find these papers.
Awesome. Yeah, I mean, and so that brings up a really great question. Like if you look at the topics on our breakout tables below, you'll see that natural history, pollinator taxonomy and natural history is listed down there. If you're interested about natural history or research here as an attendee, join that table and see why pollinator taxonomy and natural history might be an impediment to their conservation, and how we might be able to employ people's like Jess's or David's or Julian's expertise, right, to ensure that there are ways that we can overcome those impediments. So Thanks to all three of y'all for giving like super informative and engaging and interesting talks. Sure, we'll get lots more questions as well. And if folks who are attending want to drop more questions in the chat, I'll definitely reach out to our speakers and see if I can get answers back up on the chat, or when we come back together after the breakout, we can do it in a panel format. So, you know, right now I think we're right on time. I'd say for all of our attendees, you know, scroll down, look at the different topics that are listed on the breakout tables. We'll take about a half hour, maybe till 2:30 to join a table, video chat. There's a couple of questions listed in the drop-down menu at those tables, and discuss whatever's there. I'll be roaming around. Hopefully Julian, Jess, and David can also, you can just mute your camera and your microphone here on Zoom. And then if you join on the actual booth event page, you too can enter those tables and interact with people. And so I'll see you three guys back here around 2:30, and we'll continue on from there. All right. Thanks so much, guys. We'll see y'all in just a few.